So it's my honor and pleasure to introduce Judy Lieberman, old friend and colleague from our Harvard Medical School days, and she'll describe some genetic approaches to affect lymphocyte behavior and tumor biology.
Thank you. Thank you, Dane and Daryl, for organizing such a stimulating conference. I'm going to talk to you about our efforts to harness RNA interference, both for therapeutics and to learn about immunology and pathogenesis. And I have this running shoe here because what we're using are Asics, like the running shoe company, or Aptamer-siRNA chimeras. And like Hida said, he has very small antibodies, but aptamers, which are you can think of as nucleic acid antibodies, are even smaller. I'm going to show you our tumor-targeting aptamer, which is only 18 nucleotides in length, that has low nanomolar affinity. And the versatility of binding of RNA and ability to design RNAs that have multifunctional domains makes this, I think, a very attractive strategy, in addition to the fact that it's just, I'm going to show you a single RNA which we can use for targeted gene knockdown in cells that are very hard to transfect even in vitro, like lymphocytes. So this whole field really started in 2001 when Tom Tuchel showed that, first of all, that RNAi exists in mammalian cells, which we had no clue of before that, and that you could harness RNA interference if there were some way that you can get a small double-stranded RNA into the cytoplasm of cells, where it could be taken up by the RNA-induced silencing complex or RISC, which would lead to the removal of one strand, leaving a single unbound RNA, which could then seek out RNAs with complementarity, bind them, cleave them if the homology were good, and the cleaved RNA would get degraded, leading to reduced expression of the RNA. But this is a catalytic process because it would liberate a RISC complex with an unpaired RNA that could then seek and destroy additional mRNAs. And once this RNA is in the RISC complex, it's incredibly stable. We showed in our early experiments that we could get gene silencing that lasts for weeks. And now with chemical modifications of the RNA in clinical trials, there's actually gene silencing that lasts for 3 months. Because this is a very efficient catalytic process, not that many RNAs are needed inside a cell to get gene silencing. We've estimated about 1,000 based on being able to image siRNA release. And it may actually be even fewer. So if you compare that number to Avogadro's number, it's a very, very small number. So basically, there's the opportunity, if you can get siRNAs into the cytoplasm, which is no mean feat, that you could get incredibly potent and durable gene silencing. The obstacles are getting the RNA across the cell membrane. Nucleic acids are negatively charged. They don't pass through membranes. Most of the methods people have designed involve endocytosis. And then once the RNA is in the endosome, you have another obstacle of getting it out of the endosome and into the cytosol where the RISC complex lives. We've recently, last year in Nature Biotech, published a paper where we could actually visualize this process, the release, quantify it, look at the cell biology, and that was published last year in Nature Biotech. So this delivery problem has actually been solved for delivery to the liver. But to get delivery and gene knockdown in vitro in cancer cells or immune cells is still a real problem. These are the strategies that people are using for siRNA delivery. I'm not going to go into them too much. The first clinically tried approaches involve lipid nanoparticles. And in fact, the most advanced phase 3 trial, that's where the results look very promising, and use lipid nanoparticles to knock down a gene involved in amyloidosis. More recently, Alnylam, which is the leading company in this arena, and I'm on their SAB, so that's my disclosure, has been able to conjugate siRNAs to triantenary GalNAc that gets taken up by hepatocytes that have the asialoglycoprotein receptor. This is being able to be given sub-Q with amazing results. In the future, there are about 8 there's one clinical trial with lipid nanoparticles, but this strategy is being replaced now with conjugated siRNAs. But again, other conjugates people have tried really haven't worked. Even things that are high to receptors, say bound on macrophages, get taken up, but they don't get out of the endosome. So we've been, for the past 10 years or so, really focused on this alternate strategy, the Asics, the running shoes, which basically are covalent linked aptamer, linked to one strand of an siRNA, annealed to the other strand, and in cells, dicer, the endogenous RNAi enzyme, cleaves the aptamer from the siRNA, liberating it for gene knockdown. There are now there's a company founded by Larry. So aptamers are basically selected from RNA libraries with a, say, 20 or 40 or so nucleotide variable region by their binding to either a protein or a specific cell type, using combinations of positive and negative selection to get specificity. And there are now aptamers that have been raised to over 1,000 human proteins. There are a lot raised to mouse proteins. And the work we've done, we've basically picked aptamers from the literature or the patent literature and used them to our advantage. So this approach was developed by Paloma Giangrande, who's at the University of Iowa, and she linked an aptamer against the prostate surface membrane antigen to an siRNA against PLK1, the kinase that's important in mitosis. And the idea is that the aptamer binds to the cell surface receptor, gets taken up, and inside the cell gets cleaved. So we like this approach because it's simple. It's just one piece of RNA. RNAs by themselves aren't thought to be immunogenic. If you get targeted delivery, of course, that's better for a drug. You need less of it, and there are going to be fewer off-target effects. It's a small molecule, so tissue penetration isn't an issue. We've found recently, and I'll show you data about this, that we can give it sub-Q and get distal gene knockdown. For our CD4 targeting agent, we could get 80% knockdown in the spleen and distal lymph nodes. It's very flexible. You can change the aptamer to target different cells, change the gene. You can knock down basically any gene. And then you can also combine aptamers to get both, say, tumor targeting as well as function. You can link it to fluorophores, you can link it to toxins, you can link it to peptides. You can use Aptamer, like for antibodies, they're agonistic and antagonistic agents. And you could use it to not only deliver siRNAs, maybe microRNAs, mRNAs potentially. And because it's a molecule you can synthesize, you don't need a bioreactor, you don't need any, it's a lot cheaper and simpler to develop. And what we found for lymphocytes, even with an aptamer to CD4, because this is a monomeric agent, it doesn't activate the receptor, activate the cell, or change the expression of activation molecules. And we haven't looked exhaustively, but there's no obvious toxicity. So we started this with a graduate student in my lab, Lee Wheeler, who took an aptamer to CD4, which we found, and linked it covalently to an siRNA. And the idea was to see if we might be able to develop an agent like this that could block HIV transmission, that would knock down genes in all CD4 cells. And the idea was we were interested in the longevity of gene knockdown because, in principle, you could use it to create a sort of stable antiviral state. And so HIV sexual transmission is mediated by the CCR5 co-receptor. It's believed that first of all, it's actually very inefficient. It generally takes a few hundred exposures to become infected, and then only one virion manages to make its way and establish the infection of all the virions in an inoculate. And it's thought that the infection is localized to the skin, to the genital mucosa for about a week before it disseminates, providing a window of opportunity in which to intervene. And so we got involved in this with the idea that maybe could we use siRNAs as the active ingredient in a topical microbicide. And we first showed we could prevent herpes, which is an easier virus to inhibit because it infects epithelial cells that are easy to transfect. But getting knockdown in CD4 cells took us a long time to figure out. This is an in vitro experiment with primary macrophages where we're using a CD4 aptamer conjugated to a cocktail of siRNAs against 2 viral genes and the co-receptor CCR5. This one is fluorescently labeled with the red Cy3. We're comparing it to mock treatment, here no infection, or treatment with the Aptamer-siRNA directed against prostate surface antigen. And what we're looking at is the uptake of the fluorescent siRNA in these primary macrophages and HIV replication by in situ hybridization in green. This is the control where there's good infection. This is the other control with the prostate delivery. There's no delivery and no effect on infection. But in the cells that were treated with the CD4 Aptamer-siRNA, we got very good uptake in all the cells and no HIV replication. And then we used the BLT mouse that Hida told you about, the transplanted with human fetal bone marrow, liver, and thymus, which actually is a fabulous model for HIV. We get 100% infection, and the levels of viral replication mimic what happens in human, as well as really detailed elements of the immune response, including T-cell exhaustion. And we did it in collaboration with Vlad and the BLT core at the Ray Gun, led by Andy Tager and Andy Luster. And so first we looked at whether we could get silencing with these Asics. We applied intravaginally the fluorescently labeled siRNAs against either CD45 or CCR5. Looked 2 days later at CD4 or CD8 cells in the tissue and found with only picomoles of the Asics that we could get dose-dependent knockdown that was specific to the CD4 cells and not the CD8s. And it worked. The first thing we tried worked amazingly well in preventing HIV infection. Here we're comparing mock-treated mice treated with that cocktail of GAG, VIF, and CCR5 siRNAs, just with an siRNA against CCR5 or just with the antiviral siRNAs. And we're looking on the top row at the ratio CD4 to CD8 cells in the blood. And you can see all the mock-treated mice developed immunodeficiency. They developed high levels of HIV measured either by P24 GAG antigen in the blood or HIV RNA. But the mice treated with the cocktail, just CCR5 or GAG and VIF, basically were uninfected. Pretty dramatic effect. And the knockdown was sustained for about 2 weeks, as we expected. And we were able to put this in a gel. It worked even better than in PBS. And we actually were able to get even post-exposure protection with the anti-HIV gene knockdown. We didn't induce any interferons or have any side effects. And I took this slide out, but we're also able to get systemic knockdown from subcutaneous injection. So more recently, we've been using this to try to look at biology. And a number of years ago, we showed that the cytoplasmic DNA TREX1 was very important for HIV infection, that it basically enabled HIV, when it infected T cells and macrophages, not to stimulate an innate immune response. So when HIV enters the cell, the genomic RNA is reverse transcribed into an RNA-DNA hybrid and then converted to double-stranded DNA. And all of that occurs in the cytoplasm where there are sensors for viral nucleic acid. And Nan Yen in my lab asked, why aren't those nucleic acids recognized? And the key was this nuclease TREX1, which is actually the most abundant DNase in cells. And what he found was that TREX1, so reverse transcription is a very inaccurate process, and we believe there are a lot of reverse transcripts that are made that are not able to integrate into the genome, and they have to be degraded. If they're not degraded, they are recognized by DNA sensors and through the STING IRF3 pathway activate interferons, which suppresses viral replication. So it's in HIV's interest not to induce interferons. So we wanted to know, so what interferon would do in vivo is very hard to predict, because if you induce interferons recruit immune cells to the tissue. They lower the threshold for T cell activation, so they will create more cells that could potentially be infected with HIV. And so we wanted to see if we could use gene knockdown to look at what would happen if HIV, what effect would it have on transmission if we knocked down TREX1 and got a local interferon response. And this paper will come out next week. Lee designed siRNAs for TREX1, and basically it's very easy to design, starting with the Aptamer-siRNAs that'll knock down, we've knocked down FOXP3, other genes. And so first he wanted to see, would this Asic specifically knock down TREX1 in CD4 T cells in red versus CD8 T cells. It did. This is mRNA. This is protein. I got very good knockdown in vitro. If we knock down TREX1 in uninfected cells, we didn't find any interferon induction. But in the context of HIV infection, we see interferon induction. It's dependent on the pathway I showed you. And here we're just showing by imaging flow cytometry that it causes IRF3 to move into the nucleus to activate interferons. Then we looked at Radiana, who's here in the audience, developed a system for looking in human tissues at gene knockdown. So we take cervicovaginal biopsies, polarize them in agarose, and then we can apply virus, siRNAs, whatever we want, and look at least in situ in human tissue. So here, what we're looking at is the application of the CD4 Asics to human tissue, and then looking at knockdown in tissue. And you can see that in macrophages, which actually have very low amounts of CD4, we get knockdown. In CD4 T cells, we get knockdown, but not in B cells. And this was not reproducible. And TREX1 by itself in uninfected cells, knocking it down does not induce an interferon response by RT-PCR. So then what about, what happens if we knock down TREX1 in these tissues to HIV replication and interferon induction? So again, we applied the Asics twice. We like to do that just in case some cells don't pick it up the first time. Challenge with HIV, and then measure HIV replication by measuring HIV P24 antigen in the lower chamber of this system. So here are control cultures that are uninfected. There's no P24. Here are the control HIV-infected cultures. And then if we use the cocktail as before, we got suppression with two different TREX1 Asics having different siRNA sequences. One of them worked better than the other, almost completely, but not completely suppressing infection. The other a little less well. And the interferon induction was higher in the most effective knockdown of TREX1. And then just to make sure that the inhibition was actually mediated by interferons, we added blocking antibodies to interferon alpha and beta to our cultures, and saw that the inhibition was blocked by blocking the interferon antibodies, and which also blocked the interferon levels. And then in vivo, we wanted to look at what happened if we infect. These are control mice in blue that are just treated with PBS and then challenged with HIV or uninfected, or mice in which we knocked down TREX1 and then challenged with HIV. We wanted to look at the, here's the knockdown of TREX1, pretty good. And then if we look at HIV infection, this is after only 16 hours. We could detect infection in the control animals, but not in the TREX1 knockdown animals. Without HIV infection, we didn't get an induction of interferons, interferon responsive genes, or inflammatory cytokines. But with HIV infection, we got an induction of interferons in the genital tract, interferon-induced genes, but no significant increase in inflammation. And this is also IL-1 beta in the control, but not in the... Now, so what if you if you administer interferon intravenously, which has been done in HIV infection with equivocal results? There was some decrease in viral load in several trials, but no clinical, but that wasn't always reproduced. Or what if you just gave interferon intravaginally at the site of transmission? So here, this is the data I showed you just before with either the control without HIV or with HIV infection. And these are the mice that were treated either systemically with intravenous interferon or intravaginally. And with interferon induction, you do see an increase in interferons and interferon response genes. But you also see an increase in inflammation. In terms of HIV infection, actually TREX1 is an interferon-inducible gene, so it goes up in the interferon-treated mice. And there is some suppression of HIV at this early time point in the intravaginally treated interferon mice, but not in the systemic. But there's also more inflammation. So basically, most cytokines are really meant to act locally, not at a distance. And when you give the high levels you need to get local effects, you're also getting a lot of inflammation and other effects. So then we also wanted to compare what happens if we treat mice with LPS to induce interferons versus knocking down TREX1 with the Asics and then challenging with HIV. Again, we could knock down, and here we're looking at 1 and 2 days after HIV infection. So we could knock down TREX1 with the Asics. TREX1 goes up with the LPS, as I said it would. And even in the blood, TREX levels are increased with the LPS. If we look at HIV replication, it's not completely suppressed, but it's certainly a lot less than the mock-treated mice, and the LPS actually exacerbates the infection. One thing that was interesting is that in the mice treated with LPS, we saw the appearance of HIV in blood cells as early as 2 days after infection. So we actually think that it's possible in humans that have vaginal infections and inflammation that this model of containing the infection may not be accurate. So then we wanted to know what about the induction of interferons and inflammation. And so in blue, you have control animals. In red, LPS, which induces both interferons and inflammation pretty dramatically. And in the green, the TREX knockdown mice, you see that there's an induction of interferons, but not of inflammation. So that's basically what you want to do. And what about infection. So here again, we're comparing mock-treated mice infected with HIV, mice treated with that cocktail, which we know completely suppresses HIV. Here we're looking at HIV RNA in the blood, HIV P24 antigen, and CD4 T cell decline. The TREX1 knockdown suppressed the infection but didn't eliminate it, and it suppressed it with a delay of about a month, but eventually the virus took off. And I don't know if that was because we didn't continue to knock down TREX1 that we lost the knockdown. But what's interesting to me, at least, is that the mice developed the exact same level of viral set point, even though the infection was delayed and suppressed. Nobody really knows what determines this magic level. So this is sort of what we found with these humanized mice, is that if we knock down TREX1, we can activate interferons that suppress HIV and that delay transmission. But when TREX1's in the cytoplasm, IRF3 stays in the cytoplasm. There's no interferon induction, and infection proceeds. I want to just briefly tell you a little bit about the cancer work. So as I said, we have a very small aptamer, only 18 nucleotides. We can chemically synthesize these molecules. And we chose EpCAM because it's actually the first tumor antigen. It's expressed on all epithelial cancers as well as epithelial cancer stem cells. And again, as proof of concept, we're knocking down PLK1. And basically, we get knockdown of and cell death that's proportional to EpCAM expression on normal tissues, normal cells. There's no knockdown. On mesenchymal breast cancer cells in green, there's no knockdown, only in epithelial cells. And this is an aptamer that only recognizes human EpCAM, so we wanted to know, would it go to normal tissue? We took biopsies of breast cancer and adjacent normal tissue. We added fluorescent siRNAs, which you can actually see with the naked eye. And you can see the signal only goes to the tumor, not to the normal tissue. In vivo, if we put an EpCAM positive tumor on one flank and an EpCAM negative tumor on the other flank, and inject Alexa 750 labeled Asics in the scruff of the neck, we see that the Asics goes to the EpCAM positive tumor, not to the EpCAM negative tumor. So we're getting selective delivery from sub-Q injection. And this is like the anti-tumor effect. Again, the first experiment we tried, again, EpCAM positive tumor on this side, EpCAM negative on the other flank. Basically, within 10 days, complete regression. Nothing was optimized. This is just the first try. Control tumors all persisted. So we have this great way of targeting cancers. EpCAM, basically all colon, lung, prostate, and pancreatic tumors are EpCAM positive. I didn't show you this, but we basically completely eliminated tumor initiation from these tumor cell lines, and we didn't trigger any innate immunity. So what we're doing now is, so the EpCAM aptamer we have is only recognized as human, and we're interested in seeing if we can investigate immune therapy and what molecules might be good. We're interested in targeting the tumor because we'd like to avoid autoimmunity and really go for the business. And Paloma has identified a family of aptamers that were developed against rat HER2, but they actually cross-react with human, mouse, and rat HER2. This is shown here. And basically what we're beginning to look at is whether by targeting HER2 breast cancers, by targeting the tumor, we think we can look at other things besides checkpoint, which only works in a minority of tumors. Ellie Gilboa had the neat idea that if you knock down an mRNA quality control pathway, you can get the tumor to express new antigens and be recognized. We want to knock down PD-L1 and PD-L2 in the tumor. We want to knock down CD47, like Hida was talking about, the don't eat me signal for antigen presentation. And we also want to look at whether if we knock down TREX1, we might get an interferon response and increase tumor immunity. So this is just beginning work. And I'd like to stop there. Thank you.